In our last video, we're going to talk about complex patterns of inheritance. These are traits that don't necessarily follow Mendel's laws with respect to a dominant recessive pattern. The genetic basis still follows Mendel's laws, but the phenotype doesn't appear to follow them as nicely. So we're going to talk about a couple of different types, incomplete and codominance, multiple alleles, epistasis, and polygenic inheritance. The example we have here is polygenic inheritance, where we have a lot of genes all working together to produce a particular phenotype. And I find that one particularly interesting, but we will get to that. So Mendel worked with traits that showed complete dominance. This is a trait where one allele completely covers up the other. Going back to our purple and white flowers, the purple allele completely covers up the white. And in the individuals from these crosses, you are distinctly purple or distinctly white. The important thing about this is that homozygous dominant and heterozygous individuals are indistinguishable. You cannot look at an individual and say that one is the purple heterozygous because there's no phenotypic difference. With incomplete and codominance, though, the heterozygotes have a different phenotype from either of their parents. In incomplete dominance, neither allele is fully dominant over the other. And so we get this sort of blending inheritance like we saw in the first video with Mendel and the difference between blending and particulate inheritance. But the difference here is that there's no actual blending occurring. Each individual is passing on a specific allele. Those alleles produce a very specific trait. And in the homozygous individuals, you get the same phenotypes as the parents. It's the heterozygous individual that has that intermediate phenotype. So here we have red or white flowers. These are snapdragons. And in incomplete dominance, we get pink flowers produced. The reason for this is that when you have a red flower, you're getting two genes producing red pigment. When you have a white flower, you're getting no genes producing red pigment or color pigment. In the pink, in the heterozygote, you only have one gene producing pigment, and that's not enough to fully display that red phenotype. And so instead, you get a light red or a pink. So you can identify incomplete dominance by sort of an intermediate heterozygous trait, something that is somewhere in the range between the parents, but not quite either one of them. We can also have codominance. So this is different in that in codominance, both genes are displayed equally. So this means that you can easily see both types of parental traits in the individuals. So the example on the right is chicken color. So we have black chickens represented by a B allele and white chickens represented by the white allele. And in the heterozygous individuals, they are checkered chickens. They're black with some white on their body. So that white allele is being expressed in the chickens, and the black allele is being expressed in the chickens. This is different than incomplete because in incomplete dominance you would get a gray chicken, something in between. In codominance, the heterozygote shows both traits distinctively. Another example of this are the M and N blood groups. So most of us are familiar with the ABO blood typing. There are a variety of different ways you can type blood, and another way to type blood is the M and N molecules. And these are molecules that are attached to the surface of the red blood cell. If you are MM, you only have the M molecules attached. If you are NN, you only have the N molecules attached. But if you are MN, the heterozygote, you have both molecules attached to the red blood cells. This is really similar to the inheritance of the ABO blood type, and we'll talk about that in just a minute. So alleles, we've been talking about traits that have only 2 alleles, but in reality, most genes, or at least many of the genes that are encoded in a genome, have more than 2 alleles. And that makes the inheritance a little bit more complicated, because when you look at multiple alleles, you can see anything from one allele being dominant over all to several alleles being codominant, or one being more dominant than the other, and so it can get quite confusing. A really good example of multiple alleles is the ABO blood type. So there are 3 alleles for ABO blood type. There's the A allele, the B allele, and the O allele. These A, B, and O refer to antigens that are present on the outside of the red blood cell. And so if you have the A allele and you are homozygous for A, or heterozygous for the A or the O allele, you're going to have the A antigen on your blood and display the A phenotype for the A blood group. B is the same way. But A and B together are codominant. So if you're heterozygous for A and B, you have both of those antigens on the outside of your blood cells, and you're the AB phenotype. The O genotype is recessive to both. So being homozygous O is being homozygous recessive for all blood type. And in the O blood type, you actually have no antigens on the outside of your red blood cell. When we get to our lab, we'll actually work with this and see why these blood types are actually medically important. Oftentimes, a genetic disorder or a genetic trait is encoded by a single gene that has multiple phenotypic effects. So we've been talking about genes that control one aspect of the phenotype. So flower color or seed shape. And the phenotype is manifested in one particular area in that organism. But oftentimes, and particularly with genetic diseases or genetic disorders, those alleles actually have effects throughout the body. And that's because genes can be expressed differently in different types of cells, or you can have something that not only affects the immediate cell, but affects the functioning of the other cells around them. So cystic fibrosis is a good example of this. This is a disorder that causes trouble breathing, it causes weakness. Individuals are often susceptible to pneumonia. But the actual gene encodes a chloride ion transport protein. And when you have a defective chlorine ion transport protein, chlorine ions are not pumped in and out of the cell correctly, and on the outside of the cell you can get a buildup of mucus. When you have mucus buildup inside, there's problems with gas exchange, there's problems with exchange of other ions, and so not only do you have the phenotype of the dysfunctional protein, but you also have this mucus buildup. And that mucus can line your organs, it can line your airways, and cause a lot of problems with gas exchange, which is why you get a variety of phenotypic effects from this disorder. Another example is sickle cell disease. So in sickle cell disease, the red blood cells are misshapen. They're normally a nice, round, plump, almost donut type shape. And the function of red blood cells is to carry oxygen throughout the body. With sickle cell, those blood cells are not formed correctly, and they produce a sort of crescent or sickle shape cell. This limits the ability of that blood cell to move oxygen throughout the body. And so that's the immediate effect, is the misshapen blood cell that doesn't bind oxygen properly. But from there are all kinds of problems that are related to not having enough oxygen. There's muscle weakness, problems with organ function. And then on top of that, those sickle-shaped blood cells don't move through your veins and arteries very well and can actually clog up those veins and arteries, causing even more problems. So now not only are you not getting enough oxygen, you're also having problems with circulation. So pleiotropy is where we have one gene that has multiple phenotypic effects. The opposite of that is epistasis, where you have multiple genes affecting one phenotype. So in epistasis, a gene at one location affects whether or not a second gene is expressed. So a perfect example of this is coat color in Labrador retrievers. I just put a little shout out to my new puppy Scarlet. This is Scarlet and her best friend Stella. But I put her in here because these two dogs go nicely with our example. So we have two different genes here. We have the B gene that determines the coat color of the dog. So the dog is born with the genotype to either be black or to be brown or chocolate. So if they have the dominant allele, they're going to be black. If they are homozygous recessive for B, they're going to be brown. So the chocolate is recessive to the black. But there's a second gene, and that's the E gene. The E gene determines whether or not that black or brown is actually deposited into the hair follicle. So the dog might be producing pigment to be black or brown, but if it's homozygous for this E gene, that pigment's not going to be deposited. So down on the right corner, we have yellow labs who have either the brown or the black allele, but because they're homozygous recessive for the E gene, those pigments aren't put onto the hair follicle, and so the dog appears to be yellow or light colored. So in this case, even though we are doing a dihybrid cross with two heterozygous individuals, we don't get the regular 9:3 to 3:1 ratio. Instead, we get a 9:3 to 4 ratio, where more individuals than we would expect show a recessive phenotype. Finally, we get to polygenic inheritance. So in polygenic inheritance, we have, in a similar manner to epistasis, multiple genes affecting one phenotype. The difference here is that we have many genes affecting a phenotype. So more than two, often many more than two. And when we start getting interaction of a lot of genes, we get what we call quantitative characters. These are characteristics that can be measured with some sort of number value, some sort of scale, but they also vary along a continuum. So we don't necessarily have distinct categories, but instead we have a range of phenotypes. So if you look at all those skin colors down along the bottom, you have the very lightest all the way up to the darkest skin color. And that's because there are different levels of pigment that are deposited on the skin based on the number of alleles for skin color that are present. So if you look here, the light skin color is actually the most recessive, that lightest skin color. So this is recessive for all 6 of those alleles. Then we've got all these combinations between light and dark. So different numbers of dominant alleles give different levels of skin pigment. All the way up to the darkest skin color, which is dominant, homozygous dominant for all of the alleles. These genes are also called complex traits, and a complex trait is one that is not only affected by multiple genes but also affected by the environment. So your skin color can be darker if you have a lot of sun exposure. It can be lighter if you don't have as much sun exposure. Some of this can be related to nutrition. So polygenic inheritance is really interesting because it links multiple genes and the environment. And the environment actually actually plays a really important part in a lot of genes, but we see it a lot in these polygenic inheritance patterns where we have a wide range of phenotypes. A lot of our human population variation is actually due to polygenic inheritance. So hair color, eye color, skin color, height, build, all of those things that we see as a range in human populations are governed by polygenic inheritance.